Welcome to Creative Peptides. Today, we talk about growth factors. The growth of eukaryotic cells is modulated by various influences, of which growth factors are amongst the most important for many cell types. A wide range of polypeptide growth factors have been identified and more undoubtedly remain to be characterized. Factors that inhibit cell growth also exist. For example, interferons (IFNs) and tumour necrosis factor (TNF) inhibit proliferation of various cell types. Some growth factors may be classified as cytokines, for example ILs, transforming growth factor beta and colony-stimulating factors. Others, for example insulin-like growth factors, are not members of this family. Each growth factor has a mutagenic effect on a characteristic range of cells. While some such factors affect only a few cell types, most stimulate the growth of a wide range of cells. The ability of such factors to promote accelerated cellular growth and division has predictably attracted the attention of the pharmaceutical industry. The clinical potential of a range of such factors, for example to accelerate the wound healing process, is currently being assessed in various clinical trials. The wound healing process is complex and as yet not fully understood. The area of tissue damage becomes the focus of various events, often beginning with immunological and inflammatory reactions. The various cells involved in such processes, as well as additional cells at the site of the wound, also secrete various growth factors. These mutagens stimulate the growth and activation of various cell types, including fibroblasts, epithelial cells and vascular endothelial cells. Such cells advance healing by promoting processes such as granulation and subsequent epithelialization. The growth factors that appear most significant to this process include fibroblast growth factors, transforming growth factors, platelet-derived growth factor, insulin-like growth factor 1, and epidermal growth factor. Wounds can be categorized as acute or chronic. Chronic wounds, such as ulcers, occur if some influence disrupts the normal healing process. Such influences can include diabetes, malnutrition, rheumatoid arthritis and ischemia. Elderly people are particularly susceptible to developing chronic wounds, often resulting in the necessity for hospitalization. Ulceration associated with old age, diabetes, etc. remains the underlying cause of up to 50% of all amputations carried out in the USA. The insulin-like growth factors constitute a family of two closely related polypeptides, insulin-like growth factor 1 and insulin-like growth factor 2. As the names suggest, these growth factors bear a strong structural resemblance to insulin. Infusion of IFN decreases circulating levels of insulin and glucagon, increases tissue glucose uptake, and inhibits hepatic glucose export. IFNs display pluripotent activities, regulating the growth, activation, differentiation of a wide variety of cell and tissue types. The full complexity and variety of their biological activities are only now beginning to be appreciated. IFNs exhibit a wide range of growth physiological effects, all of which are explained primarily by the ability of these growth factors to stimulate cellular growth and differentiation. Virtually all mammalian cell types display surface IFN receptors. IFNs play a major stimulatory role in promoting the cell cycle. IFN activity can also contribute to sustaining the uncontrolled cell growth characteristic of cancer cells. Many transformed cells exhibit very high levels of IFN receptors, and growth of these cells can be inhibited in vitro by the addition of antibodies capable of blocking IFN receptor binding. IFN was one of the first growth factors discovered. Its existence was initially noted in the 1960s as a factor present in saliva, which could promote premature tooth eruption and eyelid opening in neonatal mice. It was first purified from urine and named urogastrone, owing to its ability to inhibit the secretion of gastric acid. IF has subsequently proved to exert a powerful mutagenic effect on many cell types, and its receptor is expressed by most cells. Its influence on endothelial cells, epithelial cells and fibroblasts is particularly noteworthy, and the skin appears to be its major physiological target. It stimulates growth of the epidermal layer. Along with several other growth factors, IF plays a role in the wound healing process. IF is synthesized mainly by monocytes and ectodermal cells, as well as by the kidney and duodenal glands. It is found in most bodily fluids, especially milk. Platelet-derived growth factor is a polypeptide growth factor which is sometimes termed osteosarcoma-derived growth factor or glioma-derived growth factor. It was first identified over 20 years ago as being the major growth factor synthesized by platelets. It is also produced by a variety of cell types. PDGF exhibits a mutagenic effect on fibroblasts, smooth muscle cells and glial cells, and exerts various additional biological activities. Fibroblast growth factors constitute a family of about 20 proteins. Typically, they display a molecular mass in the region of 18-28 kDa and induce a range of mutagenic, chemotactic and oncogenic responses. Classification as an FGF is based upon structural similarity. All display a 140 amino acid central core which is highly homologous between all family members. All FGFs also tightly bind heparin and heparin-like glycosaminoglycans found in the extracellular matrix. This property has been used to purify several such FGFs via heparin affinity chromatography. Subsequent to binding to the immobilized heparin, FGF can be eluted by inclusion of high nickel concentrations in the eluting buffer. Although many of the original members of this family stimulate the growth/development of fibroblasts, several newer members have little or no effect upon fibroblasts. Transforming growth factors represent yet another family of polypeptide mutagens. The members of this family include TGF-alpha, as well as several species of TGF-beta. If you need peptides and related services, please don't forget to contact us.